going to go ahead and get started. It gives Pramesh and I great pleasure to introduce Erika. Before I do that, though, I'm going to ask everyone, please silence your cell phones. Try to turn off your computers, etc. The lab knows we have this rule that you can't have a computer open during lab meetings, except to flip it open and look up something to prove that the person speaking is wrong. So if we see computers flip it open, then we know that Erika said something that could be wrong, which is not going to happen, of course. It's a delight to have Erika in the lab. She was born and raised in Illinois and went to the University of Illinois to Urbana and Champaign. She's joined by her family who flew here from probably sweltering heat to much nicer weather. Fair enough. Yeah.
Smoky though.
And it is smoky, yeah. Not as bad as in LA where I just came from. So, Erika has been doing a joint thesis project with Pramesh and I, and the history here is that she came and rotated through a bunch of labs and really liked both of our groups and decided to do a co-mentored project, which is very, very challenging for a student to have to deal with two mentors with different mentoring styles and very sometimes overbearing ideas about how things should be done, but it came out really good. She's been a joy to have in our groups, has taught me for sure a ton about statistics and informatics. And interestingly, you'll hear about her thesis now, which is not how it was designed originally. She was supposed to do dry lab computational work to make a discovery and then switch over to the wet lab to then validate. And the dry lab stuff was going so well that, and she was not really working on the wet lab stuff. And we realized, we sat down and said, you haven't been making any progress in the last year on the wet lab, but the dry lab's going well. Have you thought about just switching and doing only dry lab related stuff? She's like, yes, absolutely. Thank God somebody actually asked me that question. So Erika's now going to present her thesis, and then we'll have a closed session after, so I give you Erika Bongen.
Hello, thank you so much for coming to my thesis defense. I'm Erika Bongen, as advertised, and thank you for coming to my thesis, which I titled, Why Me Immunology, or Why Healthy People Have Different Disease Risks. Because the immune system has to maintain a balance, because if your immune strength is too weak, your body can be overwhelmed by a pathogen leading to infection. But if your immune response is too strong, you can have collateral damage and even autoimmunity. So I want to know, why do some people have higher risk for infection and other people have higher risks for autoimmunity? And I like to think about it this way, where genetics and the environment interact to affect your baseline immune system, leading to different risks for infection and autoimmunity. And genetics can affect your baseline immune system through a lot of different ways, through population variation, mutations you specifically have, or what chromosomes you have. Do you have a Y chromosome? How many X chromosomes do you have? An environment can affect your baseline immune system through your microbiome, or your lifestyle, diet, and exercise, and your pathogen history. You can't get shingles if you've never been exposed to chickenpox, that sort of thing. And there's a lot of work connecting genetic and environmental factors to disease risks. But I work downstream of that. I want to know what immunological factors, what genes are differentially expressed, what cell types or proportions are different between people with different risks for infection and autoimmunity. So that leads me to my thesis statement of what immunological factors give healthy people different disease risks. And I've chased after this question through two different projects. First, I examined influenza susceptibility, which got published this spring in Genome Medicine, if you want to read more into it. And my second project is on sex differences in the healthy human immune system. So first, let's focus on influenza susceptibility. Because tens of millions of people get the flu every year in the United States. And we have some obvious high-risk groups like children, the elderly, and pregnant people. But there's a good deal of heterogeneity in whether or not you'll get sick. And if you get sick, just how sick are you going to get? If we knew biological risk factors to identify these high-risk groups, then we could design new vaccines to plug these gaps in our immune systems and help people stay healthy. So the experimental setup that I've used to study influenza susceptibility is called influenza challenge studies. This is when healthy volunteers have live influenza virus sprayed up their noses, and some of them become obviously sick. They're shedding virus and they're symptomatic, so we call them symptomatic shedders. Others show no indication that they've even encountered the virus. They're asymptomatic and there's no detectable virus, so we call them asymptomatic non-shedders. So I hypothesized that the baseline immune state can predict infection status. That I can look at these volunteers before they're even infected and predict who's going to be a symptomatic shedder and who's going to be an asymptomatic non-shedder. So what data do I have to work with? So I use publicly available gene expression microarray data sets from GEO. So this tells me what genes are being used in the blood. And 3 of my cohorts, which I call challenges A, B, and C, are all in discovery. They're people infected with H3N2 and H1N1, 40 samples total, and I put them all in discovery because they're all profiled on the same Affymetrix platform, so they're technically homogeneous, and they all come from the same group at Duke. So the same people are running the same experiments. But I have my last cohort, my held-out validation cohort, GC61754, which is validation. And it has people infected with H3N2 and has 12 samples total, or 12 people total. And I put them into validation because they're profiled on an Illumina platform, so it's a completely different technology. And it comes from a group out of Oxford, so different people were running the experiment. So if I discover genes that are differentially expressed in discovery, and those genes are still differentially expressed in this held-out validation cohort from Oxford that used a different technology, that shows that the genes that I find are technologically robust, and they're robust to the group that did the experiment. So let's go find some robust genes. I obtained my data from GEO. I performed a meta-analysis across the discovery data sets. I looked for significant genes, and I tested them in the held-out validation cohort to make sure they worked. So what are these robust genes I found? Absolutely nothing. It was garbage. And I was really frustrated by this. I was ready to throw the whole project out the window. But then we figured out what was happening. The problem was I was asking too many questions with too little data. Since I was asking what genes are differentially expressed, I was basically asking 20,000 different questions because we have 20,000 different genes. And I only had 40 samples in discovery to ask those questions with, which is way too many questions for that number of people. But I'm an immunologist, and I don't actually think about the immune system on the gene level. I think about it on the cell type level, because each immune cell type is a part of an army that has its own specialty to fight pathogens. So if I can collapse these 20,000 different genes down to immune cell type proportions, that gives me a really meaningful look at the immune state of these individuals. So if I collapse these 20,000 genes down to 20 different cell types, suddenly it's meaningful information, and it's a reasonable number of questions to ask, from 40 people. Then, once I have a significant cell type, I can go back and convert that cell type back into that genetic biomarker I was looking for in the first place. Make sense? We'll go from genes to cell types, back to genes. But how do I go from genes to cell types? Well, I use a method called cell mixture deconvolution, which for the sake of time, I'm going to explain as a strawberry milkshake. So, if you're drinking a strawberry milkshake, and you know that milkshakes are made of ice cream, strawberries, and milk, and you know what each of those ingredients taste like individually, you can pretty well estimate the relative ratios of those ingredients in the milkshake, right? We can do the exact same thing with gene expression. If you have gene expression from the blood, you know what cell types are in the blood, and what they look like transcriptionally, you can estimate cell type proportions. But how do I know what cell types look like transcriptionally? Well, Francesco Bologna, a postdoc in the Kachi lab, built a tool called Immunostates that lets you do just that. He went through GEO and he found all the data sets he could that had sorted human primary immune cells. So this is not mouse cells, these are not cell lines, this is primary human. So we got 20 different cell types, 165 data sets, and over 6,000 samples. He combined all those data sets together, and for each gene, he calculated an effect size for how much that gene was expressed in B cells versus all other cell types, neutrophils versus all other cell types, T cells versus all other cell types, and so on. And we call those effect sizes immunostate scores. And if we look at an example gene, CD19, I've plotted the immunostate scores for CD19 in these immune cell types. And we can see that the one cell type with a really high immunostate score is B cells. And that makes sense because CD19 is a well-established marker of B cells. So immunostate scores tell us what cell type expresses that gene the most. And Francesco identified the 317 genes that best differentiate these 20 different immune cell types. And he used them to create the immunostate's basis matrix. So I can use this basis matrix with a linear model to estimate the immune cell type proportions in the blood. So if we know what the immune cell types look like, and we know what the gene expression in the blood for our samples, we can use the model to predict the immune cell type proportions that would be needed to get the gene expression that we observed. All right, so now that we have a way to go from genes to cell types, let's find a significant cell type. I performed a multi-cohort analysis, but this time of cell type proportions. I used cell mixture deconvolution with using immunostate to convert the gene expression to cell type proportions, performed a meta-analysis on the discovery cohorts, and I defined significant genes as genes with a false discovery rate, or FDR, less than 15%. And then I tested those significant cell types in the held-out validation cohort. And the one cell type that was significant in both discovery and validation was natural killer cells, or NK cells. And we can see this is a forest plot of effect sizes in the discovery cohorts. I mean, each of these blue boxes is an effect size, and this is the summary effect size. We can see that they're all consistently negative, showing that asymptomatic non-shedders consistently have more natural killer cells at baseline. And that's the exact same pattern we see in validation. This is a violin plot where I have plotted the estimated NK cell proportions in the asymptomatic non-shedders in blue and the symptomatic shedders in red. And each of these points is a person, and we can see that estimated NK cell proportions are significantly higher in asymptomatic non-shedders. And this is really great to know as an immunologist, because NK cells, it makes perfect sense. We know that NK cells are an antiviral immune cell type. They circulate throughout your body, trying to find virally infected cells and killing them. So having more NK cells in the blood, it would make sense that that would protect you from influenza. But can we convert NK cell signal back to gene expression? Of course we can, because I'm telling you about it. So I opened up the hood on immunostates and identified the 19 genes that immunostates uses to define natural killer cells. And then I performed a meta-analysis of only those 19 genes. So not 20,000, only 19. And I defined significant genes as genes with an effect size less than -0.5, so it had to be in the same direction as the natural killer cells, and a p-value of less than 0.05. And then I tested the genes in a held-out validation cohort. And the one cell type that fit my thresholds was KLRD1. And here is the forest plot of the effect sizes of KLRD1. Once again, we can see consistent negative effect sizes in the blue boxes and a negative yellow summary diamond, showing that KLRD1 expression is higher in asymptomatic non-shedders prior to infection. But how does KLRD1 perform in validation? It performs insanely well. I was very surprised. So KLRD1 expression is significantly higher in asymptomatic non-shedders at baseline in the validation cohort. But how well does KLRD1 separate asymptomatic non-shedders from symptomatic shedders? This is a rock plot that we use to answer that question. In a rock plot, a 90-degree angle indicates perfect separation and an AUC of 1. A diagonal line indicates random noise and an AUC of 0.5. So this is pretty close to a right angle, and it has an AUC of 0.91, which is actually a surprisingly good separator of the asymptomatic non-shedders from the symptomatic shedders. But does KLRD1 have anything to do with the symptom severity when these people are infected with influenza? Actually, it does, because KLRD1 expression at baseline negatively correlates with the log of the symptom score, indicating that the people with the lowest KLRD1 expression at baseline tend to have the worst symptoms, while the people with the highest KLRD1 expression at baseline have no symptoms at all. Now, this data was very strong, showing that KLRD1 expression is a good biomarker for influenza susceptibility, and I was willing to take a step back and look at the biology. What is KLRD1? KLRD1 is the name of a gene that encodes a protein that we call CD94. CD94 is a surface receptor found on NK cells, and it forms a heterodimer with NKG2 family members. And whether CD94 activates the NK cell or inhibits the NK cell depends on the NKG2 family member it's teamed up with. And in this example, I show CD94 teamed up with the activating NKG2 family member, NKG2C. It's recognizing an infected cell via HLA-E, and then it kills that infected cell by secreting cytotoxic granules that are full of granzymes and perforin. And since CD94 can be involved with activating NK cells and the secretion of cytotoxic granules, I hypothesized that KLRD1 expression might be correlated with the expression of cytotoxic granule-associated genes. And that's exactly what I saw. KLRD1 expression at baseline is significantly correlated with expressions of granzyme A, granzyme B, granzyme H, perforin, and CCL5, which is a chemokine that's secreted along with cytotoxic granules. And so this might indicate that if you have higher expression of KLRD1 in the blood, then you have more NK cells. And these antiviral NK cells have a lot of cytotoxic granules. So having more NK cells in the blood might mean you have more cells of cytotoxic capabilities, which is a good thing to have if you're up against influenza. But everything I've shown you thus far has been at baseline, prior to infection. What happens to KLRD1 expression during infection? So this is the pooled data from the discovery challenges. And at time point 0, these people were infected with KLRD1. And we can see that KLRD1 expression decreases in symptomatic shedders within the first 48 hours of infection. And this got me really excited, because if expression of KLRD1 is decreasing in the blood, this might be because the KLRD1 expressing NK cells are traveling from the blood to the site of infection. Now this is sort of trafficking experiment, can only be proved in mouse models, and I don't do mouse models, but the best I can do is find correlations in human data. So if I see a decrease of KLRD1 expression in the blood that matches an increase at the site of infection, then the KLRD1 expressing NK cells might be going from the blood to the site of infection. So I asked, how does KLRD1 expression change at the site of infection? And I ran into a problem, because I work with public data, and there's no publicly available data sets of KLRD1 expression in the nasal epithelium. But we came up with a solution, because Marta Andres-Terray in the lab found a shared transcriptional signature of viral infection. She pulled data sets of influenza, respiratory syncytial virus, and human rhinovirus data sets together, and used our multi-cohort analysis framework to identify a transcriptional signature of the host response against viruses. And since the host response against influenza, RSV, and HRV seems to be shared, then KLRD1 expression might not just be important for influenza, it might also be important for HRV and RSV. So I hypothesized that KLRD1 expression increases in the nasal epithelium of people infected with HRV and RSV. That's exactly what I saw. This is gene expression data from the nasal epithelium of people infected with HRV, RSV, and RSV co-infected with other pathogens. And this is roughly 48 hours post-infection, but sometimes it's hard to count that with natural infection. And we can see consistent positive effect sizes for rhinovirus and RSV, showing that KLRD1 expression does increase at the site of infection during HRV or RSV infection. And what's really, really cool is that one of these data sets is actually a rhinovirus challenge study. So they have an early time point, 8 hours post-infection. And this time point is so early that the original authors didn't discover anything interesting happening 8 hours post-infection. But I found something interesting. Because KLRD1 expression 8 hours post-infection negatively correlates with the log of the symptom score, showing that the people with the highest KLRD1 expression post-infection have barely any symptoms, and people with the lowest KLRD1 expression 8 hours post-infection have the worst symptoms. Now this data supports a model where a rapid response by KLRD1 expressing NK cells can reduce your symptom severity with rhinovirus infection. So to conclude the influenza challenge portion of my talk, I found that asymptomatic non-shedders have higher expression of KLRD1 in the blood, and this is most likely because they have higher proportions of natural killer cells. And my data supports a model where a rapid response by these KLRD1 expressing NK cells is sufficient to eliminate the infection rapidly and making you an asymptomatic non-shedder, or it at least reduces your symptom severity. So now I've shown you how NK cell proportions are involved with influenza susceptibility. Let's take a different angle on disease susceptibility. Let's look at sex differences in disease risk. Because you remember that I said that if your immune response is too weak, you're at high risk for infection, but if your immune response is too strong, you're at risk for autoimmunity. There's a lot of data showing that males are biased towards infection and responding too weakly against stimulus, while females are biased towards over-responding and ending up with autoimmunity. We can see this in epidemiology, where the majority of autoimmune diseases predominantly affect females, such as systemic lupus erythematosus, which affects 9 women for every 1 man. But we also see this in infection, where men in the United States are more likely to die of this parasitic and infectious disease. And so to fit this into the model I showed you before, we look at how genetics and environment affect the baseline immune system. But when we're talking about what's different between human men and women, we don't talk about genetics and the environment. We talk about sex and gender. Sex is what's biologically different between males and females. It's what chromosomes you have, it's your hormonal milieu, and it's the fact that females evolved to support and protect the fetus. Gender is the activities and behaviors that society defines as masculine or feminine. And it can affect your baseline immune system through your environmental exposures. Most construction workers are men, most nurses are women. This will put different stressors on your body and on your immune system. And society also places different values on diet and exercise for men and women. And diet and exercise affects your microbiota, which then affects your immune system. So sex and gender interact to affect your baseline immune system, giving you different risks for infection and autoimmunity. And once again, I'm interested downstream of sex and gender. What's different between the healthy male and the healthy female immune system that leads to such different disease risks? And that's a pretty huge question. So I need to limit it down and figure out how to ask it. So I decided to work with gene expression microarray data sets because gene expression shows us both cell type proportions and immune activation. I decided to focus on healthy humans, and here I define healthy as people with no active infection, no autoimmunity, and no cancer. And I focused only on humans between the ages of 18 and 40 years old because I wanted to study the population post-puberty and premenopause. And for tissue, I decided to use white blood cells, and that means both whole blood and peripheral blood mononuclear cells, or PBMCs. And I chose to look at the blood because it's the most readily accessible immune tissue, which means I have access to data sets from all over the world. And if I use data from all over the world, it means that the genes that I find are more likely to work for everybody. So I found 16 data sets that fit my inclusion criteria. 6 are in discovery, and I have worldwide coverage with Australia, China, Morocco, UK, and the United States, about 50-50 whole blood and PBMC, with 447 samples total. In validation, I have 10 data sets from Australia, Finland, Netherlands, Denmark, South Africa, Singapore, and the United States, with 524 samples total. You'll note that I have a lot more data to work with this time around, so I don't have to go from genes to cell types and then back to genes. I can go straight to genes. So I can find robust genes using multi-cohort analysis. So I performed a meta-analysis in the discovery cohorts, and I defined significant genes as genes with a false discovery rate, or FDR, less than 5%. That means that statistically speaking, 5% of my genes will be false positives. And I set an effect size threshold of 0.4. Now this is a moderate effect size. And I chose a moderate effect size because men and women aren't different species. The differences between us aren't really that huge. But it's the adding up of these many small differences that can lead to differences in disease risks. Okay. And that allowed me to find the immune sex expression signature, or ISEX, which is 144 genes. 94 genes are higher expressed in females, and 50 genes are higher expressed in males. And then I tested those genes in the held-out validation cohort. And here's what the ISEX genes look like. So these are heat maps of the effect sizes, and you can think of effect size as fold change of the I-sex genes in the discovery and the validation cohorts. Orange indicates a positive effect size for genes that are higher expressed in females, while purple indicates a negative effect size for genes that are higher expressed in males. And first you can notice there's very consistent purple, very consistent orange, indicating that these genes are differentially expressed consistently across both discovery and validation. But what's even more interesting is I put a color code up here for the chromosomal location of the I-sex genes. And we can see that the genes with the darkest purple, the genes that are highest expressed in males compared to females, these are all on the Y chromosome, which makes sense. Females don't have that chromosome, they can't express those genes, so of course males express them more. That's a nice biological sanity check. And on the other end, we can look at the genes that are highest expressed in females compared to males. And these are all known X-escape genes. That means that these genes are known to be expressed by not just one, but both of the X chromosomes. Since females have 2 X chromosomes and males only have 1, females tend to express twice the amount of mRNA that males do. So once again, a biological sanity check showing that our methods work. But what I found really interesting is that 75% of the I-sex genes are colored gray, 75% are on autosomes, which are non-sex chromosomes. And I wanted to separate the effects from the sex chromosomes from the autosomes. So I put the genes into 2 different buckets. If a gene was on the X or the Y chromosome, I put it into the sex chromosomal bucket. If it wasn't, I put it into the autosomal gene bucket. And then I collapsed these buckets down to individual scores, the XY I-sex score and the autosomal I-sex score. And I calculated these scores as the geometric mean of the female-associated genes minus the geometric mean of the male-associated genes. This means the higher your score, the more female-like your expression. The lower your score, the more male-like your expression. Okay? So how well do these scores separate males from females? They both work very well. If you remember, this is a rock plot that tells you how well a score separates 2 groups. And a 90-degree angle is perfection, and a diagonal line is random chance, random noise. And you'll see that the XY I-sex score is nearly perfect with a summary AOC of 0.99. And that makes total sense, because the definition of sex, the definition of male versus female, is what chromosomes you have. So of course, if you look at the sex chromosomes, you'll be able to differentiate males from females. What's more interesting is how well the autosomal I-sex score works. Because here, we have a summary AOC of 0.76, which is significantly better than random chance, but not a perfect separator. This indicates that the autosomal I-sex score captures nuanced but robust sex difference information in the healthy human immune system. Okay. But so far, all this validation I've shown you has been on the same type of technology. It's been on gene expression microarrays. So does I-sex work on a different type of technology? To answer this question, I went to the milieu interior cohort, which profiled gene expression of 1,000 healthy volunteers. And to save money, they used a technology called nanostring. So they only looked at the expression of 560 hand-picked immune-related genes. And of these 560 genes, only 13 were present on I-sex. But I hypothesized that if I-sex actually captured true information, we'd see the same sort of effect sizes in these 13 genes in the milieu interior cohort as we did in discovery. And that's exactly what I see. This is a scatter plot where each point is a gene. On the x-axis, we have the I-sex discovery effect sizes, and on the y-axis, we have the effect sizes from the milieu interior cohort. In orange, we have female-associated I-sex genes. These are genes higher expressed in females. And we can see that the effect sizes, they're all positive in discovery, and they're all positive in a milieu cohort, indicating that they're all tend to be higher expressed in females. What's even better is the purple genes that are higher expressed in males. Because not only are all the effect sizes negative in the milieu interior cohort, but in fact, some of them are even stronger in the milieu interior cohort than in discovery. So this is great. This shows us that I-sex works in different technologies. But so far, I've only shown you that I-sex works in XX females and XY males. The human race is incredibly diverse, and there's a lot more of us out there. So how well does I-sex work in XXY males? which is called Klinefelter syndrome. And it's actually a fairly common phenomenon which happens in 1 in every 501,000 males. And it's thought to be underdiagnosed because the symptoms of it aren't all that severe. They tend to have higher estrogens than typical males and lower androgens, and they have a female-like risk for systemic lupus erythematosus. So I hypothesized that both the XY isex and the autosomal isex scores will become more female-like in Klinefelter syndrome. And that's exactly what I see. This is a box plot of the XY isex score in typical females, Klinefelter's males, and typical males. We can see that Klinefelter's males have significantly higher XY isex score than typical males, but it's still lower than typical females, most likely due to the Y chromosome gene expression. Okay? But what about the autosomal isex score? So, once again, the Klinefelter's males have significantly higher score than the typical males, so they are significantly more female-like. But this time, the Klinefelter's males are indistinguishable from the typical females, which is a really exciting finding because typical females and Klinefelter's males both have the same risk for systemic lupus erythematosus. So it's possible that the autosomal isex score may capture some biological factors that affect our risks for autoimmunity. So far, I've shown you that isex works across technologies, it works across Klinefelters, typical females, typical males, and so it works. Like, we can move on and ask some more scientific questions. And as an immunologist, what I'm really interested in is immune cell types. So what immune cell types express isex genes the most? And if you remember from the beginning of my talk, how Francesco Bologna built immunostates, he combined 165 data sets with over 6,000 samples and calculated immunostates effect sizes, which tell you how much, it tells you what cell types express a gene the most, such as CD19 is highly expressed in these cells. So if I look at the immunostates effect sizes for isex genes, I can identify what cell types are driving isex. So first, let's look at the female-associated isex genes, the genes that are highest expressed in females. And here, orange indicates a high isex score, not knowing this. It's a high immunostate score, showing that that gene is highly expressed in that cell type. And purple shows that it's not. And here, the cell type of interest is CD4 positive T cells. And I point them out because you can see a consistent orange all the way across, showing that CD4 positive T cells highly express the vast majority of female-associated isex genes. But what about the male-associated isex genes? Well, it looks like this. And the clusters I'd like to highlight are macrophages, monocytes, basophils, and neutrophils. And since all of these cell types are myeloid cells, it indicates that the male-associated isex genes are highly expressed on myeloid cells. But these are a lot of myeloid cells, and I actually don't think that all of them are driving isex. And the reason I don't think that is because not all of them exist in the tissues that I study. Monocytes, macrophages, because macrophages, neutrophils, and basophils are mostly in tissue, but some of them can be found in whole blood. And the trouble is, I studied isex in both whole blood and PBMCs. So whatever myeloid cells is going to be important has to exist in both whole blood and PBMCs. And the only myeloid cell that does is monocytes. So most likely, monocytes are the cell type driving the male-associated isex genes. But not everyone in this room is an immunologist. So what do CD4 T cells and monocytes do? So if you remember, the female-associated isex genes are highly expressed by CD4 T cells, and CD4 T cells are a fantastic coordinator of immune responses. CD4 T cells are part of your adaptive immune system, which means they remember pathogens that you've encountered before. And if a CD4 T cell is shown a microbial peptide from that pathogen by an antigen-presenting cell, it will activate and secrete cytokines in order to coordinate an immune response specific to that pathogen. But what about monocytes? Monocytes highly express the genes from the male-associated isex score. So monocytes circulate in the blood, but when they leave and they go to tissue, they hulk out and become macrophages, the big eaters of the immune system. And macrophages devour dead cells, but they also devour pathogens like viruses and bacteria. So macrophages are really good first responders. So the female signal seems to be coming from the coordinator, the male signal seems to be coming from the first responders. So next I asked, if CD4 T cells and monocytes are so important, are they driving ISEX due to cell proportions? And the reason I ask this is because I work with blood gene expression. And if a gene is higher expressed in the blood, it could be because the cell type that expresses it is expressing more of it, or because there's just more of that cell type. And it can be tricky to tease this apart, but I can do that by correlating flow cytometry, which directionally measures immune cell proportions, with gene expression. So let's look at the data. This is a scatter plot where each point is a person. Red points are female, blue points are males. And on the x-axis, we have the naive CD4 T cell percentage measured by flow cytometry. On the y-axis, we have the autosomal ISEX score. And we see a non-significant positive correlation. And it's in the direction we expect because the female-associated ISEX genes are highly expressed by CD4 T cells, but it's not statistically significant. What about monocytes? Monocytes correlate in the same way. So the monocyte percentage significantly correlates negatively with the autosomal ISEX score, with an R of minus 0.79, indicating that the people with the highest monocyte percentage tend to be males and tend to have the lowest autosomal ISEX score. And so this shows me that monocytes are major drivers of the autosomal ISEX. So now that I know that ISEX works, I know what cell types drive it, I can ask even more interesting questions. I can ask, how does ISEX change during an immune response? And I chose to study influenza because influenza fits nicely into the paradigm where males have weak responses and females have two strong responses. And we know this because males are at higher risk for a poor flu vaccine response, and females are at higher risk for severe flu infection where you end up in the hospital due to excessive immune responses. So I asked, how does ISEX change during an influenza infection? And once again, this is my influenza challenge data. And we have the autosomal ISEX score on the y-axis and time in hours on the x-axis, and red is for females, blue is for males. And at time point 0, these individuals were infected with influenza. Now we can see the females start out significantly higher than the males, but then they dive down to male-like levels about 48 hours post-infection. But around 100 hours post-infection, which is about 4 days after infection, they spring back up almost higher than they were in the beginning. And when I saw this, I was like, oh, this is a really cool pattern. I bet it's due to differences in monocyte proportions, because I had that nice monocyte correlation. It has to be monocytes. And so I performed cell mixture deconvolution to get the estimated monocyte proportions. And I looked in these samples, how do estimated monocyte proportions change with infection? And we see that both the males and females start out about the same at time point 0 when they're infected. The females may get higher than the males, and faster than the males, around 48 hours, but I have small sample size, so I don't know if that's significant. And then they go down at the same rate, around 100 hours. So this pattern is very different from the autosomal ISEX score pattern, so I don't think the monocytes could be driving the autosomal ISEX pattern I observe here. So what about the CD4 T cells? Well, it looks exactly the same. So females start out high at time point 0, they dive down and become indistinguishable from the males around 48 hours, and they spring back up higher and faster than the males around 100 hours, indicating that the autosomal ISEX score is detecting sex differences in the dynamics of the CD4 T cell proportions during infection. And I found this really exciting because then the autosomal ISEX score can capture sex differences in the dynamics of the antiviral immune response. And we need to do more work, but this could help explain why males and females have different outcomes with infection. But the active immune response is only one part of the immune response. The immune system also has to build memory against that infection. So can the baseline autosomal ISEX predict antibody response to influenza? And I have one cohort that measured antibody responses to a naturally acquired infection. Now this cohort comes from Texas A&M, and at the beginning of the school year, they recruited undergrads, and they asked them to come back within 48 hours of getting a flu-like symptoms. And then they had the undergrads come back at the end of the school year to get the post-flu measurement. And they measured their antibody titers against flu at the beginning of the school year and at the end of the school year. And so by measuring the change in the antibody titers, we can measure the strength of their antibody response. So I asked, does the autosomal ISEC score correlate with antibody responses to influenza? And I hypothesized that since females tend to have stronger immune responses, that the more female-like your autosomal ISEC score, the stronger your antibody response. And that was exactly what I saw in males. So we have a significant correlation between your change in antibody titers and the autosomal ISEC score, showing that the males with the highest autosomal ISEC score, who have almost like female-like levels of autosomal ISEC score, at the beginning of the school year, end up having the strongest antibody responses when they get infected that flu season. But there is no significant correlation, no relationship I can detect between the baseline autosomal ISEC score and your change in titers for females. And if you use the baseline autosomal ISEC score to predict who's going to be a high responder versus a low responder, you have a very good AUC for males with an AUC of 0.92, but for females, it's not statistically significant from random chance. And this is actually really exciting for males because when I compare the baseline autosomal ISEC score in males to the spring autosomal ISEC score, it's incredibly consistent. The males who are high at the beginning of the school year are the same males that are high at the end of the school year. Quiet, Kim. And this could actually be useful because if this stays consistent, not just over 1 year, but over years and years and even decades, you could conceivably make this be a test at the doctor's office where the doctor runs your blood. It's like, oh, you're at high risk of being a low antibody responder. We better put you on higher dose vaccines that will keep you better protected. But what about the females? Because the autosomal ISEC score just didn't work at all for females. Why could that be? And I can think of two possibilities. One is that the autosomal ISEC score just isn't informative for females. It captures some aspect of the male immune system that just isn't the way females do things. But the other option that I want to look into is menstrual cycle. Because I only have the autosomal ISEC score at the beginning of the school year. And if the autosomal ISEC score is responsive to hormones, like I suspect, and it fluctuates, then I really need to know the autosomal ISEC score on the day that female was infected. Okay? And I see a little bit of this when I correlate the baseline autosomal ISEC score with the spring autosomal ISEC score. For the most part, we see a similar correlation in females. It is statistically significant, where females who have high autosomal ISEC scores at the beginning of the school year also have high autosomal ISEC scores at the end of the school year. But there are some discrepancies that I find really interesting. Like if we look at these two females, they have almost the same autosomal ISEC score at the beginning of the school year, roughly 0.4. But at the end of the school year, she shoots up above 1 and this female shoots up below -1. What happened to them to make them so different? One possibility is menstrual cycle. Fortunately, I don't have any data on menstrual cycle. So if the future directions I would do if I had time to do another PhD would be to look at menstrual cycle, which luckily my friend Emily is looking at. So hopefully we'll have data in a few years. Sorry to put you on the spot. So, in conclusion, I showed you how I identified the ISEC score, which is a signature of consistent genes that are differentially expressed between the healthy male and female immune systems. I showed you how I traced these cells back to stronger monocyte signal in males and stronger CD4 T cell signal in females. Hopefully the ISECs can help us better understand why males are at high risk for infection while females are at high risk for autoimmunity. And hopefully it can tell us about infection because I've shown that the autosomal ISEC score correlates with antibody response, but only in males. And I've shown that males and females have different CD4 T cell dynamics during influenza infection. And maybe it can even tell us about autoimmunity because Klinefelter's males, who have the same risk for lupus that typical females do, have indistinguishable autosomal ISEC scores from typical females. So, in conclusion, all my work trying to figure out why healthy people have different disease risks seems to boil down to cell types. where asymptomatic non-shedders have more natural killer cells at baseline than symptomatic shedders, and males have more monocyte signal, while females have more CD4 T cell signal. And this is actually really good news to me because as a field, immunology has so many new ways to look at immune cell types. We have CyTOF, which can measure more cell types than we ever have been able to. We have cell mixture deconvolution, so that we can repurpose old data to find cell type proportions. And we have single-cell RNA-seq, which will help us find even more cell type subsets. And I think we'll be able to better understand immune system disease susceptibility in the future. So I want to thank everyone who's helped me on this project, everyone in Pravesh Khatri's lab, especially Francesca Blania for building immunostates, and Wynn Haynes for helping me stay sane, and Marta Andres Ture because her work on influenza infection really paved the way for my project on influenza susceptibility. I also have to thank everyone in PJ's lab for ordering wonderful snacks, and Kelly McGill for great discussions on sex differences. And thank you so much to my committee, especially Parag for being there since the beginning. And thank you, Marcia, for teaching me everything I know about sex differences. And Marcia runs the Stanford Wisdom Center, the Women and Sex Differences in Medicine. So if any of you are interested in sex differences, join the Wisdom Center or go talk to them because they will help get you started. And do you have any questions?